गुड आफ्टरनून सर।
सर वेरी गुड आफ्टरनून।
सर मेहता सर कौन सी आईडी से ज्वाइन करेंगे? डू यू हैव एनी आइडिया?
नहीं सर, ऐसी तो कोई बात नहीं हुई।
अच्छा, उनकी आईडी बता देना एक बार।
मेहता सर
वो-
की।
हाँ, उनको होस्ट बनाना पड़ेगा ना, नहीं तो प्रेजेंट नहीं कर पाएंगे।
ओके।
हाँ, एक
एक
बार
बार वो
आप।
लिंक से ज्वाइन कर लेते हैं सर, उसके बाद जैसे कर लेते हैं हम।
हाँ, वही पता चल जाएगा ना हमें कि वो कौन से हैं तो।
ओके।
हाँ, एक बार उनसे व्हाट्सएप से पूछ लो चाहे।
ठीक है सर। Hello, Dr. Smith.
So, good afternoon, all, and let me welcome our guest, Professor S.K. Mehta, for today's lecture. And as we are already 5 minutes late, so I would not like to waste more time for all introduction of this our lecture series. So, I welcome our Professor Chairperson, ma'am, to start the proceedings. Thank you, ma'am.
Thank you, Dr. Sumit, for inviting me. Welcome, Professor Mehta. Namaskar. And good afternoon to all of you. And I feel my proud privilege that today I am welcoming such an eminent professor as well as renowned researcher, not only in India as well as in abroad also in many countries. And it I feel quite fortunate enough for myself as well as for my department and my students also to have Professor S.K. Mehta among us. And we will we will be benefited from his lecture and his discussion with him. As far as I know about his research profile and his achievements, later on, Dr. Krishna Kumar will give a formal introduction about Professor Mehta, but I can feel proud to aware you about his major achievements. Like Professor Mehta has H index of 49, which is a very big achievement, with I-10 index 208, citations 8507, and is an author of about 20 books or book chapters. He has been nominated through various projects, research projects in India as well as abroad, in collaborations also. And moreover, he has guided so far 47 PhD students, and still some postdoctoral fellows are benefited by his guidance, as well as he has guided 50 master students for various projects. Moreover, I again feel proud to speak about his achievements, that he has completed or completing 5 DST, 5 CSIR, 6 UGC, 2 Indo-German, and 1 Indo-Japan research or exchange projects in present times and in past also. So I would like to say my students that you listen the invited lecture with very patience, and you can clarify their doubts after the lecture is over. If you are not able to join the talk through verbal discussion, then you can type your message in the message box, chat box. And now, not wasting much time, I would like to invite Dr. Krishna Kumar, which is also one of the organizers of this lecture series, to give a formal introduction about Professor Mehta. Thank you, sir. Thank you very much.
Sir, namaskar. Good afternoon, everyone. I heartily welcome one and all present here. A warm welcome to our distinguished speaker. We are honored to have with us today Professor Surendra Kumar Mehta, which is a senior most professor in the Department of Chemistry, Punjab University, Chandigarh, and he was also ex-director of SAF, CIL, UCIM, and is highly active in the significant areas of research like metallosurfactant chemistry, nano-electrochemical sensors, synthesis and application of semiconducting nanoparticles, and colloidal chemistry that includes novel formation of the drug delivery applications. He is also coordinator of Chandigarh Region Innovation Knowledge Cluster, CRIKC, local coordinator MHRD Initiative Gyan, and coordinator UGC CAS. So Professor Mehta has more than 350 publications in International Journal of Repute with H index of 49 and I-10 index 208. having citations 8507, and is an author of about 20 books. He has been nominated as a member of several DST, CSIR, and UGC national committees. He is also a recipient of the renowned DARD and JSPS fellowships several times, bronze medal from Chemical Research Society of India, CRSI, and he is also a fellow of Royal Society of Chemistry, UK, Haryana Vigyan Ratan Award, and Professor W.U. Malik Memorial Award of the Indian Council of Chemists for the year 2015. For his outstanding contribution in research, he has been a visiting scientist to several countries like UK, Germany, Japan, USA, and France. So Professor Mehta has guided 10 postdoctoral, 47 PhD, and 50 master students. He has completed 5 DST, 5 CSIR, and 6 UGC, 2 Indo-German, and 1 Indo-Japan research exchange projects. So this is the brief introduction of Professor Mehta. और सर की जैसे हम कंप्लीट इंट्रोडक्शन अगर देना चाहेंगे तो मुझे लगता है सर पूरा दिन ही हमें लग जाएगा आपकी इंट्रोडक्शन बताने में. So with these words, I would like to invite Professor Surendra Mehta to deliver his talk. Mehta sir, please.
Thank you. So first of all, I would like to thank both the chairperson, Professor Suman Lataji, and Krishan Kumar, Dr. Krishan Kumar, for such a elaborative and nice introduction and all the praise they have showered on me. Thank you very much from the core of my heart. Now, I was supposed to deliver lecture last year also, but somehow I could not because of circumstances. Today also there was a problem, but then I thought I have to deliver whatever the circumstances. Yesterday, my wife, she got this coronavirus. She is positive, and you can understand I became so busy in household affairs. So I thought first I should decline, but then I thought no problem. I will take care of that, all these things, circumstances. And so I'm here to deliver the talk today. And sorry for a little late. I was trying to connect, but the link was missing somehow. I missed that. Anyhow.
Sir, 1 minute, pardon. Sir, if you are comfortable, then please continue. Otherwise, it is okay. Please.
Okay, it's okay. It's okay. Now she is sleeping and she is taking rest, so I can take that. No problem.
Sir, please, if you are convenient, only then we. Okay,
no
okay,
problem.
sir. Thank you, sir. Thank you very much.
It's a question of mindset. If you prepare yourself in that direction, I think it can be done. So I thought I should go for this lecture. Anyhow, so you can all of you realize that we are living in a very bad situation. This corona has created havoc, in particular in our country, and every third person is getting infection. So I don't know what will happen, but anyhow, the people are brave enough to tackle all these issues. But although we have been taking utmost care, but still you never know where from you can caught this virus. Anyhow, so let me share my PPT and then we start discussing. Will you allow me to share, or just directly I put on this my PPT?
Sir, please, you can share.
I just click on now. Is it visible? Not yet.
No, sir. अभी तो visible नहीं हुई sir screen आपकी.
Okay. So sharing का देख लेते हैं. Is there any sign coming? I don't see that.
Sir, there is a sign of share screen in green.
Okay, okay, yeah.
Yeah, yes, sir. You can.
Okay, I think now it must be visible.
Yes, sir. Yes, sir.
Okay.
Sir, with your due permission, can we record this meeting?
Sure, sure. Go ahead.
Thank you, sir. Thank you very much.
Nothing to hide. Okay. So the title is very broad, because we have been dealing with nanochemistry or nanotechnology, as you can see, that I thought broadly describing some of the systems which are useful for sensing, catalysis, and drug delivery. Although this is quite broad, but then I thought it is the best to actually, it is the motive is that we have to encourage the students and researchers to do their best in this field, because this field is very exciting and a lot can be done because of the existing infrastructure which is available nowadays in universities and otherwise also at national level. There are some good laboratories where all the infrastructure is available, and you can utilize those, because to do this type of research you need high-end instrumentation, and as you mentioned in your in the introduction that I was the director of the SAP Center. These are sophisticated analytical instrumentation facilities, which these centers are there. 15 centers all over India, and Chandigarh Punjab University is having one of them. So DST gives a lot of funds to establish high instrumentation techniques, because in this research you need, for example, SEM, TEM, XRD, and whatnot. There are so many instrumentation techniques which are available, can be utilized very effectively for doing nanochemistry research. So at Punjab University, we are fortunate that we have this type of facility. I know that at your place also you have established already one of such laboratories, and more instruments will be coming. I keep on getting feedback from Professor Sharma. I think he will be heading the center at your place. And but you are near to Delhi also, so that is the advantage that in Delhi there are so many institutes where such facilities are available, and students and teachers can approach there to get their sample analyzed. Moreover, these 15 centers are the SAP centers are paid facility, and if you want to get your sample analyzed, you have to pay some amount and send the samples, and you can get the results depending on how much time they take. Some facilities they take less time, some they take more time. So there is a portal called Srishti. Sorry, the Srishti portal is created by DST, and this is shared link for all the SAP centers. And once you click the Srishti portal on DST website, information about all the 15 centers you will see. And you can choose which instruments you need and which place it is available. And accordingly, you can contact that. And there's most of the places like at Chandigarh, there's online submission facility. You just pay online and send your sample by post. And within no time, you can get the results. So this type of facilities they're there, and I think students and researchers can benefit that. So this is just to tell you about this thing. Another thing which was mentioned in my biodata was that this CRIC, Chandigarh Region Innovation Knowledge Cluster. Just I would like to mention about CRIC, because you know that in Chandigarh and in and around Chandigarh, like Mohali and Panchkula and up to Ropar, there are so many institutes which have come up. So in Chandigarh, we have Punjab University, for example, we have NIPER, we have IMTech, we have CIAB and INST, ICER. So there are so many institutes in the, particularly in Mohali. And similarly in Panchkula, we have TDRL, DRDO, and and many other institutes. And there are so many private universities in and around Chandigarh. So taking, this, our previous Vice Chancellor had this idea that all the institutes or even some of the companies, they should come and make a cluster. So this cluster is nothing but Chandigarh Region Innovation Knowledge Cluster. The advantage of forming this cluster was that we are able to use the facilities of other institutes, and other institutes, for example, can utilize the facilities of Punjab University. And library facilities we can use, even interaction between students, interaction between researchers, joint programs for seminar, symposia, or for even teaching of classes. For example, if some teachers are not available in a particular field, we can invite teachers from CRIC institutes. So that way this CRIC cluster was formed. It's very much appreciated even at national level. And this is a very famous cluster where we are continuously interacting with the other institutes. So I think this type of cluster can be formed in every city, in every place, like in Murthal, if you have other universities or institutes or even colleges, you can make a cluster, and continuous interaction, meetings, seminars naturally are benefit to the students and researchers also. And the place where less research is there, they can be encouraged to do more and more research. They can be involved. The facilities can be available. No doubt, these facilities can be available at nominal charges, but the charges can be same for all the institutes. So this type of ideas, I have been the coordinator of that, and we have done a lot of work in this, and it has become very famous cluster. And people are really taking benefit of that. And we invite for the membership of that also, and people are becoming member also, individual members also, institutional members also. So this is going on very well. Taking advantage of this, or the idea was noticed by the central government, and they have created Pune cluster, for example. They have created Delhi cluster now. They have created a Mumbai cluster, so Hyderabad cluster. So there are many clusters are coming up, and this is just because there should be cooperation. Otherwise the facilities which are available in one institutes are not available to the other institutes, researchers, and sometimes they are underutilized also. So idea is that you have the facilities available for everyone, and the maximum output should be generated out of those facilities available. So this type of ideas was there. Anyhow, coming forward, so I mentioned about the COVID situation. Just a minute. Yeah. So I will start my lecture with the SARS-CoV-2, which is COVID-19, and is creating very havoc. Why I'm mentioning this other than my own work? The reason is that only some time back, I asked my student to explore if people are using nanotechnology for finding remedies for COVID-19. So she came up with some data which I would like to show before I go to my own work. So my question was, can nanotechnology and material science help the fight against SARS-CoV-2? Because nanotechnology, as you know, everywhere it is being used, in every field it is being used, and why not tackle the COVID-19 situation? And this is a brief overview that how this is affecting everybody. We have a coronavirus. This looks like this, and this is called SARS-CoV-2, which is basically severe acute respiratory syndrome. And it is 2. Severe acute respiratory syndrome, which is popularly known as COVID-19, and it was detected in December 19. Now this virus, which is there, I think many of you must be knowing, because a lot of information is available on the internet and also newspapers, and we have been discussing many lectures. You must have, listened. Now, what is the size? The question arises: what is the size of this coronavirus? You know, this. If you look at the literature, there are fluctuating reports, but I found that 50 to 140, we say that this is the total size which we have. This virus has. So its size is very small. You can't see that. That's why you can't see that, because it's very small size. And you see on this spherical shape particle, there are some brown areas. You can see these are some looks like tumors. You know, we have some tumors on this, and the size of these are 9 to 12 nanometer. So this is a total coronavirus, that is COVID-19 particularly, and which is creating havoc in our society. You know, now other things are, you know, that it is, it can be transmitted through your mouth, through your eyes, and so you keep on washing your hands. You use gloves. Hands should be hygienic, and now report says that it is present in the form of droplets in the air also. So the situation is very, very alarming, you know. And the droplets, you know, when we speak, the droplets come out of our speech, and these droplets having, what is the size then? The size is 5 to 10 micrometer. The droplet size which we generate out of our mouth when we speak are in the range of 5 to 10 micrometer. So size is large as compared to when we say it is 50 to 140 nanometer size, but droplets, they have higher sizes. So these droplets contain so many large number of 50 to 140 nanometer SARS COVID-2, CoV-2. So now, you know, that's why the importance of masks come into play. So that's why WHO and government also time to time telling that we should wear mask, otherwise, you know, these droplets can transmit COVID-19 to you also. When somebody in front of us is speaking and he's producing these droplets, and they can be transmitted if we don't have any protection on our mouth and nose. So therefore, putting mask is very important, and nowadays we say that we should wear a double mask, you know, so that virus should not go inside through our mouth or nose. So even use of goggles, use of mask, even this PPE kits, you know, when it's all for the protection, these are done. And you have nanoparticle based drugs coming into the market, which are hybrid nanoparticle drugs, and a lot of research is going on. So this is what my student found, that there are many references where nanoparticles have been used to understand how they can be useful to prevent this coronavirus. So I'll not go detail. This slide I'm showing you just to tell you that people are working in this area, and they are trying to use the knowledge of nanoparticles, which we understand clearly, and because of a lot of work available, a lot of methods available in the literature, how to prepare different types of particles, and these particles have been used for different purposes. For example, functional carbon quantum dots. This is a very, very important paper. I will show you. I will show you a couple of papers also. Functional carbon quantum dots as medical countermeasures to human coronavirus. Very important paper. Somebody has published. And similarly, people have used cationic carbon dots based on curcumin, which is basically antiviral. Then we have this gold nanoparticle people have used again for the colorimetric assay to detect, because detection is also very important. Now RT-PCR tests are being done, rapid tests are being done. So we need methods where quickly we can find out. So colorimetric method is also one type of method where gold nanoparticles are being used. And label-free colorimetric assay has been done. Then we have multiplex paper-based colorimetric DNA sensor, using silver nanoparticles. So again, this is for use for virus only, and rapid and sensitive detection of anti-SARS-CoV-2 IgG using lanthanide doped nanoparticles. So some publications are there where they have used lanthanide doped nanoparticles. Then silver sulfide nanoclusters, the glutathione capped AG2S nanoclusters, again for viral RNA synthesis and budding. Then we have delirium BSA nanostars. Then we have gold nanoparticles. Then gold nanoparticle modified carbon electrodes. People have used electrochemical immunosensors. So biosensors are also very important. Biopolymeric nanomicrospheres also have been done. Dendritic cell targeted chitosan nanoparticles. They are biologically accepted. So they have been used. Silver nanoparticles people are using. And there are many, so many publications are there. Polymeric nanoparticles, they have prepared and again used for respiratory syndrome coronavirus. Another very important was this copper-based additives, and particularly for hospital environment, which are, we know that copper is very much used for antimicrobial behavior, and then antimicrobial polymers have been used for this type of purpose, and copper oxide particles are being used. So you can see a lot of work is going on, and why not here? Everybody can do that. So this is just a idea because of this pandemic situation. Our society should also, researchers, scientists should also contribute in this area. So one of the paper which was very important, I thought I should mention that. This is functional carbon quantum dots at medical countermeasures to human coronaviruses, right? So in this work, they use 7 different carbon quantum dots. I'll show the figure, that's better. So you can see, now this is your coronavirus, and we have a receptor here, which is the main problematic. This receptor is very problematic because this is when virus goes into the body, and this it operates through this receptor, and it tries to enter into the cell membrane. And that's why we say that it is going into the lungs, and it is damaging, and it is multiplying, and it is creating problem for the lungs. So this penetrates the receptor, through this receptor penetrates into the body, and it goes to the cell membrane, and then the damage starts. So this is a problem. So this study which was done was very interesting. That basically idea was to make this receptor inactive. So once we make this inactive, naturally it will not penetrate into the cell membrane, and we are safe. There is no problem. And how to make this non-active? And this was done by using quantum dots, nanoparticles. And here they used functionalized quantum dots, and this functionalized quantum dots was basically derived from 4-amino-phenyl boronic acid. So 4-amino-phenyl boronic acid. So this quantum dots was created, and they created many of the quantum dots by different functionalization. Like here they have, they use this acid and created another type of quantum dots, which is they called 5. This is quantum dot 6. Then they saw that this quantum dots, they interact with this receptor and make this receptor inactive. So that was the idea. So because, in this virus, we have proteins. And these proteins, they interact with the quantum dots, and therefore they will make this inactive. So this is a very interesting study. So I thought this should be mentioned, that they prepared hydrothermal carbonization. Using hydrothermal carbonization, these quantum dots were prepared, and they binded with this virus to cells, and so many types of quantum dots they created. Quantum dots 1, quantum dots 2, 4, 3. By functionalizing, they use citric acid, methyldiamine, and just could functionalize ascorbic acid, copper sulfate, and so on. So you can see different functionalization was done. A detailed study was done, and for the quantum dots from 1 to 4, which are shown here, they use colorimetric method for the detection of the coronavirus. So this you can see, this is in water, this is in this PBS, and then this was the prepared medium, which was prepared, and they saw that there is a color change. So colorimetric change was there, and using different quantum dots, color was slightly different. They measured the particle size also, and you can see, and how the particle size also varies, and the distribution is different. So this is a 5, quantum dot 5, quantum dot 6, 7, and so on. Again, colors you can see, there is a colorimetric change is there, and using TEM you can see the size. Another interesting was the manufacturing process of antimicrobial critical medical devices, and they have used antimicrobial polymer. So this is because now we require so many medical devices, which should be antimicrobial, and where we can use antimicrobial polymer. And the process starts with this corn, which they have taken, and taking this corn, they used they created lactic acid, lactide, then using this having polymerization, polylactic acid, PLA, and then you can convert this into the pellets. This polymer pellets are there now, but it is having antimicrobial properties. So you can convert this into antimicrobial filament, and using 3D printing, you can create critical medical devices. So these are very, very important nowadays. So many companies have come up in India because of the encouragement of government, that we should produce our medical, we should not depend on the China, rather we should produce medical devices in our country only. The advantage is you can recycle also, because they have been created from the environment friendly material. So you can also recycle it after use, and you can again convert into pellets, and again convert into filament, and using 3D printing again, you have medical devices. So here we are also using this copper nanocomposite additives, because when using this copper nanocomposite, and your system becomes antimicrobial. So there comes the role of nanotechnology, nanochemistry. Another very interesting is the novel anti-influenza copper oxide containing respiratory phase mask. And this, it's not a very new finding, but it's 2010, and Boroku and Zhu and others, they studied this. That time they were not aware of that this will be so useful nowadays in present situation. But because in hospitals also doctors and nurses and other staff, they use this mask, So the idea, I think that was the idea, to create a mask and having some coating, so it should be antiviral, antibacterial, and the use of copper oxide particles is very important. So this type of mask they proposed. This is in this paper. They proposed that this type of face mask should be there, and that's why we can save us from the virus and other infection pathogens. And here, they proposed 4 layers. This is the outer layer, and this is the inner layer, D, A, B, C, D. There are 4 phases in this. And they have outer, 2 external spun-bound polypropylene layers, A and D. You can see A and D, these 2. And it contains 2.2% copper oxide particles, weight by weight. And it also has polypropylene layer, and which contains 2% copper oxide particles. So you have spun-bound polypropylene layer, and then you have the metal blown polypropylene layer, which contains 2% copper oxide nanoparticles. And then you have also polyester layer containing no particles. So this was the proposal, which nowadays has become very useful. I will also tell you about our experience in this. So this way you can easily protect, because when virus will come in contact with your copper oxide particles, automatically it will die. It will interact with the particles, and its receptor will automatically become inactive, and there is no chance it can go inside your mouth or nose. So they have shown this external layer, internal layer, and external layer is thick thread type, and this is also thread but thin, and you can see the presence of copper. One can see easily from the SEM photographs, and on the layers you have clear copper oxide particles. So this is very useful. Now, although we have seen this paper later, but before that, during the last lockdown, because we have been working a lot in this nanochemistry or nanotechnology, so one of my students, he was quite interested. So we together, although there was a lockdown, laboratories was closed, but somehow we tried to make a formulation. So idea was to have this type of formulation coated on the mask, so that we can protect ourselves completely. Even if virus comes closer to the mask outer layer, it should not affect. Because otherwise, there is a possibility if mask is not proper, it can penetrate through the mask, and it goes into the mouth and your nose and create a problem. So we were successful in having this type of formulation. We used some polymers and copper nanoparticles and silver nanoparticles, and we had an agreement with the Benicia Biotech. They showed interest, and they said, okay, we will produce the mask and where this coating can be done. So we gave this type of information to them, and even we gained also some consultancy was given to us, and they are in the process of testing, and I think it may come into the market soon. Besides that, we thought, why not to test this type of formulation for other purposes, And for surfaces, for example, like you have your mobile phone, why not to cover this mobile phone, which basically we are using day and night. It can have some infection, the particles can be on the surface, or we are using computer, we are sitting on the table, chair. If we are able to have this formulation, which we can put on this on these appliances or in the surrounding, then it can certainly protect us from coronavirus. So with that idea, we did many experiments from ICER, also biological testing we got done from a private lab. We got done all the testing, biological testing, and other SEM, whatever possible, whatever is required. And this formulation is very effective, and it can protect any, if it's put on the surface for 90 days, it protects us. So now, my student, although he's doing PhD, but he, in the beginning, he has this startup company in his name. So he is trying to sell this to startup, and this has come on the in the market also with the name of Vox, VOX. So it is available on Amazon, and if you on Amazon, if you write VOX, so all information comes, and NanoVox, the name is given, and the information is available, and this formulation is also available. Recently, one of the hospital, they invited, and they got spray there, and I think they are happy that the results are positive. Okay, so this is just I wanted to tell you. So coming to what we have been doing, so this I will just skip because there are some more references also, interesting ones. Already a lot of time is there. So a few examples I will give you how we started this, and how some research can be done, although many of you are already working in this area and knows ins and outs of the nanoparticle fabrication. So one of the interesting was because I have been working right from the beginning of my career in colloidal chemistry. So colloidal chemistry, I have been dealing with surfactants, all type of surfactants. And we have been, in the beginning, we used to create systems, bicellular systems, and we moved to microemulsion system. We also created solid triple nanoparticles and so on. And a lot of work initially we did. And later on, as most of us, attractive towards the nanochemistry. So I was no exception. Also, I thought why not to go towards the nanochemistry also. But our base was the surfactant chemistry only. So we saw that surfactant chemistry can be utilized effectively in the nanochemistry. So for example, if we functionalized our surfactants, or we introduce metal in the surfactant, and then we call this as metal surface active agents, and these are popularly known as metallosurfactants. And we use these metallosurfactants for creation of the nanoparticles. So we found that these are very effective nanoreactors. You don't, because in nanoparticle formation, always you require a sorry, I'm missing that. Sorry, capping agent. So always we require a capping agent, because otherwise agglomeration takes place, and you cannot have a particular size of particles. The size increases if the agglomeration takes place. So you always require a capping agent. But while using metallosurfactants and creating nanoparticles, you don't require any capping agent, because already surfactant is there. So automatically it will, and metal is there, surfactant is there. So it will convert into a metal in the nano range, and metallic nanoparticles will be created, and it will be on the surface. You will have the surfactant coated, and therefore a stable nanoparticle will be formed. And the second example I can take, this microwave irradiation method is very, very effective. And we created a lot of nanoparticles where we used microwave irradiation method. Then low temperature synthesis, homogenization, microemulsion using plant extracts we also try to use, and for different purposes we can use for drug delivery, for as catalyst, for dye removal. A lot of work is being done on dye removal, fuel cells, and as sensors. Okay. So the idea was very simple. We can have this metallosurfactant. We can create self-assembly of these metallosurfactants, or otherwise you can have use the method of thermal decomposition, or even you can use the method of reduction. You can also do liquid crystal templating. All these procedures can be done. So we preferred the self-assembly, and where you can have create the self-assembly of these metallosurfactants, and then you can convert into the nanoparticles. So the idea was very simple. I have just, for our students, I'll briefly tell the basic what idea was. So synthesis was carried out in water heptane by basic redox system, and very simple process we did. We had the metal acetate we have taken and dodecylamine, and we dissolved in N-heptane, and we N-heptane phase using sonication, and you then use sodium borohydride, and slowly you can add, and for 2 hours, and then you allow this bi-basic solution for equilibrium, and polar and non-polar phases separate. So you'll have, because you have polar water is there, and you have heptane, so bipolar system is there, bi-basic system is there. So nanoparticle can be extracted from either, whether they are in heptane region or whether they are in water region. So depending on which particle is in which region, you can separate them. So it's a simple process, as you can see. So metal is affected in heptane. You are using sodium borohydride. So we have prepared copper, nickel, cobalt nanoparticle, which you can see in N-heptane, whereas iron, zinc, they are in water. So easily you can, this bi-basic solution is there. You can see easily the color change will easily tell you the particles have been formed, nanoparticles have been formed, and you can separate because it's a bi-basic system. And usual systems are characterized. I need not to tell you that FTIR is initial analysis done. Then U visible tells us if there is some peak at some wavelength. So that indication is that you have the particle formation. For example, copper, you have almost 580 or between 80 and 90. One peak, you will observe U visible peak at this wavelength, and similarly in nickel you have around 300. In iron you have a very small peak at 300 something, 370, 310, 605, and here there are many peaks in the zinc, and so that is already known in the literature. There is nothing special. You can easily find out. So, and IR will tell you which components are there in which are coating the nanoparticles, and also you can easily see that you have dodecylamine pure, which we have used, and where we have zinc introduced, where we have iron introduced, and you can differentiate. And similarly, you have dodecylamine, copper, cobalt, nickel. So these are initial techniques which can be done. Particle size measurements can be done. Some you can see very small particle size, 1 to 2, and it goes to 13 to 14 nanoparticles. Size nanoparticles are found. So again, this can be easily done. So metallosurfactant, as you can see, by using these, you can create the quantum dots. There's the advantage of that. So you have very small 1 to 2 nanometer size, 3 to 6, 7 to 8, very small. So there is one advantage of that. The other is microwave irradiation. Microwave, I need not to tell that the microwave method has become very, very famous because it shortened the reaction time by a factor of approximately 20. Heating is uniformly spread. There is no problem. throughout the entire bulk of the reaction mixture. Then you have also this method is very important because you can get narrow distribution of particles. So that's a very advantages we have for this. So one example, there are many examples we have, but only one example I'll give you. We have metal precursor you can take, and for environmentally friendly system, ruthenium nanoparticle, for example, you can use glucose. You have PEG 20,000, and just put all the material in the metal nano. You can create the nanoparticles, and you can also see the variation temperature with time using a software. How the temperature varies inside. Very, very important. So maximum temperature has gone to 87 degree in 82 seconds. The process is done in very small time, and you can monitor. So you can have the effect of microwave heating time. You can have effect of glucose, effect of PEG. So all these variations are done. You want to optimize your system for getting the best information. That how much time it will take to get the minimum time is required for the getting the good particles, and how much glucose is required. And we have varied from 7 millimole to 28 millimole, PEG from 0.062 millimole to 0.375, and we can optimize this very well. In this, you can see if you are using 25 watt for 8 minutes or 100 watt for 2 minutes, 200 watt for 1 minute. So if you use 200 watt in 1 minute, you can get the particles, very nice particles, small particles. But if you are having 25 watt, then you require more time, that is 8 minutes you require, and particle size also slightly bigger particles as compared to when you have 200 watt 1 minute. And you can see there, if you, the sample A, B, C, we have compared in the table. You can, from the 10, if you see, the particle size is from 2.2 to 5, right? So if you are having 200 watt 1 minute, so you have 2.2, and the highest one is 5 nanometer for A when you have 25 watt. And from DLS, you get 3 to 7. So there's not much difference. This difference is always permissible. So again, you can see you have created quantum dots using microwave, very simple method. Just mix all the chemicals and use the power to get the particles. So same procedure have been done for various types: zinc sulfide, palladium gold, palladium ruthenium, and so on. Again, I'm giving you a very simple method initially, the synthesis of zinc oxide nanoparticle. I think many methods are available. Simplest are available. So zinc acetate you take and have sodium hydroxide and just 90 degree, stirring at 90 degree for 30 minutes, you'll get zinc oxide nanoparticles. Characterize them as usual using PEM, XRD, and so. And you can also create iron oxide nanoparticles. So one of the students initially did all that way back in 2011, 13, 14. He was doing PhD. But I will show you interesting part. So he could create these particles very easily. And again, having Teflon autoclave nowadays, this is a very popular method. You have the autoclave and you can create the nanoparticles in autoclave very easily. So characterize them and you can find the morphology and other things. So then we have using the plant extracts. That method is very popular. Environment-based methods are also very useful nowadays. So here we use globe extract or ginger extract, CE and GE. So which are easily available even in our houses. These materials are available. So you can get the extract by following this procedure. First you ground it, then you boil it, and then you can collect the superintendent and then label this C, E, and G. So you have to first characterize this what are the components available, and the procedure is sonication, solvent preparation, and finally you get the nanoparticle if you are using polymer plus solvent plus surfactant. And so this procedure one can easily use, but the main thing is that you can use various drug molecules, biomolecules, and you can incorporate in these glove extracts or ginger extracts, and this PLGA nanoparticle which we are preparing from C and G nanoparticles, and you can use anti-cancer biomolecule loaded on these particles, PLGA particles, because PLGA particles have been most accepted ones for use in the body. So a lot of work has been done on this, and two types of these drugs were encapsulated, and we saw their release also, and their pictures you can see. The particle size 141 nanometer, 151 nanometer, and then characterization of both KCE and NCE was done. And again, the particle size was found. It is between 100 to 200 nanometer, which is acceptable from biological point of view. You can also create nanoparticles, which is chitosan. Chitosan nanoparticles are also very good one. So you use here sodium tripolyphosphate, and chitosan you take and in acetic acid, and using homogenizer and centrifugation and lyophilization, you can get the chitosan nanoparticles. Chitosan nanoparticles are also very important from biological point of view. They are acceptable up to certain concentration range, and they are not toxic, and therefore one can use these for biological purposes. We also use few years back this warm microemulsion method to create solid lipid nanoparticles, where we use oleic acid as lipid base, which is oleic acid is liquid lipid, and the solid lipid we use is surfactant DDAB. So which we can range 12, 14, 16, 18, all types of is available. And aqueous phase tween 80, again very much acceptable from biological point of view, ethanol, water. So we prepare liquid phase, we prepare aqueous phase, and we can mix it. We call this hot microemulsion method, where we keep 75 degrees temperature, and then we freeze it, or not freeze it, but we use cold water for 0 to 4 degree. And using these two steps, we can create solid lipid nanoparticles. So spherical nanoparticles, as you can see, with cationic lipid and trap inside the non-ionic emulsifier shell, which stabilizes the particles against the coagulation and coalescence. So you can see the particle size and SEM and AFM, all these measurements were done. And this is the proposed structure which we have. We have the lipid matrix inside, and surfactant is coated outside along with the co-surfactant. And you have along with that this OA and oleic acid and DDAB inside as the liquid matrix. So it is stabilized by surfactant. That is, so tween we have using, and also the alcohols which we are using, that's co-surfactant. So one of my students went to Germany and did continued this work, which we did in our laboratory here, creating nanoparticles, solid nanoparticles. And she did a small angle neutron scattering analysis in Germany. She stayed there for more than 1 year, and she could visit these facilities there, nuclear reactor facilities, and she did a lot of work on that. So this is a typical graph which we obtained, having I versus intensity versus vector, and Q is basically the vector in this analysis. And you can have a Q range which is low, you can have high Q range, and you can see all the parameters. So many parameters are there: radius, molecular weight, scattering invariant, surfactant surface, and so on. So all these parameters for different types of DDAB, which can be used, and she used all this, and she used this molecular weight for aggregates MW on this, depending on I, that is intensity, density, Avogadro number, and other such parameters which have been used. So she could try for all the pure components first, and then finally she varied even the tween 80 from 0.1% to 3.6%, and there in the second B part, all the tween 80 in water, DDAB in D2O, tween 80 in D2O, all combinations she tried to find out. And finally, it was found that this DDAB is trapped inside the emulsifier shell. This was proved by scattering analysis. And the excess of tween 80, that's polysorbate, in the system is leading to the formation of ellipsoidal micelles. So this ellipsoidal micelles, which we could find that they are formed, this cannot be found from other techniques like TEM, SEM also, they don't tell us clearly about this. So the scattering techniques are very useful. They can tell us about the exact shape of the systems. So basically the scattering techniques are based on the models. So you create the scattering data, and then this is fitted into the various models. So you can have a model for spherical particles. You can have a model for non-spherical particle cylinders, for example, other types. And therefore, you take the data, this scattering data, and you fit in the models. So where you get the best fit, that means and after analysis, this fit tells us what type of shape is you are getting. So this is about all the data: number, density of the micelles, volume of ellipsoidal micelles, number, density, radius of spheres, and all these parameters for different types of DDAB she found. So see, this is the overall picture she could develop. That initially we have spherical nanolipid particles, but as we move further, concentration increases, you have ellipsoidal micelles. So why finding is very important, because when you are dealing with the system, what type of particle shape you require, or what type of particle size you require, you can manipulate your system if you know the prior information that we have the micelle formation or this type of shape formation using this type of concentration, and that can be very useful to you. So these are model fittings which I just skipped. Besides that, we have also worked on metal chalcogenide nanostructures for third generation device applications, where metal chalcogenide nanostructures with enhanced properties is the requirement, and here we can use the twin base surfactants which were used, which are called Gemini surfactants, which can be used, and we can vary we can create the controlled shape and size structures. So you can see that we have a spacer in between two head groups, having this tail, two hydrophilic head groups, two hydrophobic tails, and you have a spacer in between. And this is a single one where you don't have spacing. So shape controlled synthesis can be done, and you can create 0 dimension, you can create 1 dimension, 2 dimension, 3 dimension particles, which will have different shapes. As you can see, the shapes are different from others. So you can create different shapes. That's why we call them as 0 dimension, 1 dimension, 2 dimension, and 3 dimension. And these are having different applications. Like 0 dimension, they have bio application, photocatalysis, heavy ion people are using for these. 1 dimensions, they are nano electronics, nano optoelectronics, sensors, batteries. two-dimension electrochromics, cosmetics, catalysis, solid lubricants. Third dimensions, mainly they are used for medical purposes, environment cleaning, renewable energy, and so on. So depending on our requirement, our interests, you can create different methods are available. You can modify that method as per your needs and so on. So all types of systems were created: copper telluride, copper selenide, silver telluride, zinc telluride, copper telluride, again different types, and you have lead telluride also. So, important parameters which were used, we used hydrothermal method, we used a temperature which varied between 100 to 150, and time was 4 to 32 hours depending on the particles which we are creating. We have metal precursors, range of the 0.5 to 2 millimole. Reducing agents, we are using stabilizing agent, and depending on CMC, we can use it. So all types of particles can be generated. Nanostructures can be generated using different methods. So some interesting things which we saw that this work, most of the work was done by one of the postdocs. She did postdoc with me, and she created so many of these particles, nanostructures. So this table shows you copper telluride, the size range. You have metal precursors, which we have used, metal acetate, sodium telluride. You have used hydrogen hydrate as the reducing agent, and then we have stabilizing agent, which was used at temperature 120, time 8 hours, and we got the shape. Now you can see the shape can change if we change the parameters. You have more, you have same here, but you can create the copper the telluride sizes 130 by changing the system. You can also create a spherical nanoparticle. Here you get the rods, So here we have used the temperature 80 degree and time 8 hours, and we are using BSA and hydrazine as the same as reducing agent, and you can get the rods. So you can play around and to create different types of different shapes of the nanostructures. Lead telluride, nanocubes, silver telluride, hollow rods. You can create zinc telluride. You can create hexagonal bipyramids. Zinc telluride also you can have bipyramidal rods. So you can vary the components. You can vary the time. You can vary the temperature, and you can manipulate. See how beautiful pictures can be generated. And similarly, size also is there. So as I told you, these which were created can be used for various applications, although we are not doing all the applications, but some of these we are focusing. And but this is a literature, if you see, these are very, very important, and they can be used for third generation device applications. So we also, because zinc oxide nanoparticles, as I told you, have been created. A lot of methods are there. A lot of people have created that, but inside understanding is not available in the literature. So we thought, why not to discuss about that? So we wanted to see what is the effect of heating rate during the thermal decomposition of zinc acetate on the morphology of the nanostructure zinc oxide, right? So simple decomposition we can understand here. You can see zinc acetate having some water. So first step is to remove the water. We will create zinc acetate, and the next step, in the next step, the loss is there, and you get the zinc oxide. Carbon dioxide will go, and zinc oxide formation. So this is a step how this takes place. So we can say that you heat in the first step, you get zinc acetate. Rearrangement takes place. In the third step, you have zinc oxide formation, and finally you have carbon dioxide which goes on. So this is a mechanism that can understand when you have this takes place. So we try to see that how the release of acetic acid changes the local pH acidic, and we wanted to find out the reason. So we found that favors the zinc oxide nanorod formation. So once release of acetic acid makes the local pH acidic, which favors the zinc oxide nanorod formation. That's why nanorod is formed. So this is XRD. You can again compare one with each other and find out how different from one another. So this is our this thing, and if I zinc oxide, and if I heat it at 40 degree per hour, you can get this. If I do 80 degree per hour, I can get this here. If I do 120 degree, I get this here. And 200 degree, you get this here. So these are some micrographs. You can see that presence of sheet-like morphology of 200 micrometer size. You can see 200 micrometer size, which on calcination leads to the formation of nanorods and nanoparticles. So the zinc oxide at 40 degrees centigrade shows the formation of nanorods. The size is 390 nanometer to 25 nanometer. When the heating raise is increased, size of 70 nanometer, 65 nanometer, and 55 nanometer, along with the nanorods in the range of 275 into 30 to 30 into 30 to 110 to 30 was observed. Okay, so this one can easily understand that. So it seems that heating rate, although it does not affect the diameter of the nanorods, but the length of the nanorods considerably decreased from 390 nanometer to 210 nanometer, along with the formation of additional nanoparticles. This was the conclusion which was made. So this is an interesting study to understand how zinc oxide particles behave. Similar study was done on tin oxide nanoparticles and combination of these two, tin oxide, tin sulfide heterostructures. Because when you form a heterostructure, then how the shape changes, how the particle size changes, that we wanted to understand. So tin oxide has spherical particles that we know, and we have studied also, having particles 15 to 20 nanometer, but having a sulfur source in this, that is in the form of tin sulfide. Hexagonal sheets of tin sulfide also appeared, in addition to spherical particles of tin oxide. Because tin sulfide has hexagonal sheets like morphology. So both you can see in the when you have together heterostructure, you can see both in the particles. So HR10 was taken, and it shows some data. Lattice spacing was 0.3 nanometer and 0.18 nanometer. So it shows that this micrograph shows that we have a firm interface which enables smooth and especially available interfacial electron transfer, and which is very useful for photocatalytic activity, because we use this for photocatalytic activity. Zinc oxide also, tin oxide and tin sulfide also together. Another interesting which was done, still work is going on. One of my students tried to form this stimuli responsive hydrogel composite materials for potential environmental and biological applications. So this is a very interesting study, because although you see a lot of work is being done for dye degradation, but their nanoparticles are being used and hydrogels are being used. But only thing is, if you want to reuse the particles, then you need a system where you can recover easily and then again you can use it. So for this, we try to form a design and synthesize a magnetic iron oxide incorporated chitosan graphene oxide hydrogel. So we created a hydrogel which was chitosan graphene oxide hydrogel, which was created, and in situ reaction was taken, in situ mineralization, and we incorporated iron, magnetic iron oxide inside it in situ. So we get iron ions loaded this hydrogen nanocomposites. So this is CSGO hydrogen, which is having iron oxide. So I mean, this can be created if you use sodium hydroxide for this purpose, and you can create this CSGO IO. IO is nothing but iron oxide nanocomposites. So when you use this prepared nanocomposite for the effective removal of the cationic dye, for example, methyl blue, a lot of work has been done. Many studies are available. From aqueous solution was investigated by performing a series of batch absorption experiments. So we found that this hydrogel nanocomposites were found to possess excellent adsorbent property. One thing, even after 4 successive cycles at different pH of the solution. So it provides us cost-effective material for dye removal applications. The other thing is, how we reuse it. Once we do the process, we can do the magnetic separation. Using a magnet, you can separate the iron particles. And once you separate, you can again do the dye degradation. So using the same particle. So 4 times this was done, and we found that it is working extensively. Although this hydrogel is also being used for other purposes. So this is a micrograph of the FSM of the nanocomposites with iron and without iron. So you can see this differentiation. So the figure shows that we have formation of random aggregation where distribution is distorted fashion in the hydrogen. In hydrogen, it is distorted. But once we have used the iron here in the second graph picture, then you have a more compact packing of the nanostructures. So which is responsible for better performance. So that is polymeric hydrogel matrix is much more organized. So this is a comparison of the TEM, because we wanted to see what size also. So we have a 2D structure, and we have transparent sheet uniformly dispersed in iron oxide nanoparticles inside the hybrid hydrogel matrix. So a very interesting study. It is already published also. So I am not showing that. We are also trying to use this type of hydrogel based system for medical purposes, for in medical purposes, for wound healing. So that results also is under process. Another interesting study one of the students has done. She went to UK under Commonwealth Fellowship, and she used this green synthesis of molybdenum oxide nanoparticle. We studied this work here, and then she completed there also, and which was decorated but with the carbonaceous mesoporous composite with catalytic and absorption properties. So here, because that institute green chemistry is there. I mean, this is a green in the University of York Institute for Green Work only is there, and they are working in this type of area. So the idea was to have biomass utilization. So the polysaccharide source was taken, and using microwave at 4 degree 48 hours, you can create polysaccharide gel, And further on, you can dry, you have solvent exchange. Then this polysaccharide, you can create carbonaceous material. And this carbonaceous material, which is called, because of the hydrophobicity, it is called starbons. So you have in starbons, you have various functional groups, as you can see in the table. And these materials are very stable, and they are being used for tunable porous materials. They have high mesoporosity, they have high surface area, you have controllable electrical property, and because of such properties, they are very, very useful. So she created in using microwave synthesis and taking ammonium heptamolybdate nanoparticles, and in acidic environment, she could create pure molybdenum trioxide hexagonal nanoparticles, nanorods. And along with it, carbonaceous material, which was biomass derived corn starch, because as in the previous I've shown you that he used corn starch. But we found that the BET surface area is only 0.8 meters square per gram. But when we have carbonization, that is carbonaceous material is prepared, which is mesoporous, you have surface area 332 milli square per gram. So as you can see, the surface area increases very high. So these two in combination can be easily used, and then this two are mixed with starch, and it acts as a template for growth of nanoparticles, which restricts the size also. And this is how from TEM and SEM graphs tell us. You have microwave gelation, retrogelation process was done, solvent exchange, freeze drying. Finally, you have pyrolysis can be done. And first one will give you pure expanded starch with particles of 75 to create from biomass, and then the pure starbon, which was created, which is having this BET surface area 368 meters square per gram. And you can see even the SEM also shows very nice porous material. So this is again mixing of both cornstarch and the second one AHM, which was prepared, and you can again do the pyrolysis, and you get the expanded starch. You have the dopant, carbonaceous dopant, when you have the molybdenum material particles put in this. So this is a dopant one, and here the SEM also you can see, which shows more surface area in this. So nanoparticle embedded carbonaceous mesoporous composite is fabricated, which can be used for further absorption and catalytic reduction of harmful pollutants. She targeted PNB and successfully done this. Please stop me if time is already 4:30. You can stop me whenever you like. Okay, I will give one or two examples, and I think we can discuss. See, evaluation of nano assemblies. This work is also going on in our laboratory for the encapsulation of immunomodulating agents. So, one of our organic colleague, they are creating immunomodulating agents. They are called TLRs. I will show you in the next slide. And but solubility is a problem, and there are so many other problems. So we can create nano assemblies in which our lab has been working, and we can encapsulate these immunomodulating agents, and then they can be used effectively for different purposes. So that was the idea. Even many pharmaceutical molecules also, they show poor performance because of the various reasons. They are not soluble, there is no control release, and so on. So we can create these nano assemblies, and in this context, we have been working on nanosomes. Nanosomes are, although this picture you see, there are, drugs can be incorporated in various assemblies. You have nanomulsions, we have solid lipid nanoparticles, nanolipid crystals, nanocrystals, and polymeric nanoparticles. Various nanoassemblies have been used in the literature. We also can use nanosomes. They are being created using non-ionic surfactants. So basically, you have two layers, and you have the hydrophilic component. You have polar head facing hydrophilic region, and the hydrophobic component also. And this can be easily created in the lab. So, and drug then can be incorporated, and we also did all that. So BBIQ is one drug, and IMT is another drug they are producing. BBIQ is a TLRR, which we call agonistic, and DIMT is IDO inhibitor, and these can be easily incorporated in the niosomes. So they are providing us these agonistics and ITO inhibitors, and we prepare these formulations. So we found that we could achieve high entrapment efficiency, and then in vitro release also was very good. And location and partition coefficient of the drug loaded in niosomes, we could evaluate. So these are release studies. One can see how slowly the drug is released, because ultimate purpose is to send the drug in the form of this formulation in the body, so that there is a release, but slow release, and effective, the drug can be used effectively and for longer time. So these are the method how we have used, and Triton X100, PEG 20,000, and so on we have done. So now let me know how much time more, so that accordingly I can finish my presentation. Please. How much
Sir,
time?
if you feel like to continue, then you are most welcome. But if you feel you should wind up, so again you are welcome, sir.
Okay, I'll take 5-10 minutes. So
Okay, thank you.
Quickly I can. Okay, now as for the applications, as I told you in the beginning, we can use some of these for as catalysts, fuel cells, sensors, and drug delivery. So I will skip these 5 nanoparticles as catalysts. So we used these metallosurfactants for understanding the very famous this reaction, where we can convert nitrophenol to aminophenol, although this reaction is very well known. But as such, it is not, it doesn't go, but if you use the nanoparticles, then nicely you can understand simply by UV visible how this reaction can be nitrophenol to aminophenol. So we also tried this reaction using these nanoparticles, which we created from metallosurfactants. And what we did, we modified all the parameters, how we wanted to see how much time it will take, how much when the reaction is carried out in the presence. But we found that aminophenol can act also as a poison or a quencher, and it slows down the reaction. So we wanted to see how much fast reaction which is going on, till how much time it will stop or it will hinder. So we found that. So microwave synthesis of ruthenium that we use for dye degradation. And here we use this Congo red dye, and we found that we can degrade this dye simply by measurement of the UV visible. You can see initially you have a very prominent peak. With the time, the peak disappears, and the color also changes. Color is now colorless. So we analyze how this Congo red, in the presence of this catalyst, how this is broken into different molecules, and this we identify this molecule in the basis of mass spectra also. We also try to see how much initial concentration of dye is required, what's the reaction time, what is the rate constant. So all these parameters one can easily study and optimize. So, how much effect of the nanoparticle concentration, how much ruthenium nanoparticles we should use. So all these different studies we also did. So this was basically the mechanism which was proposed, that ruthenium nanoparticles are acting as a mediator in the ion transfer from the borohydrate ion to the azo bonds. So this was the reason how this degradation takes place. Then we also did some work on the fuel cells, and again I'll skip this. So we used this for understanding the having the fuels as formic acid, methanol, and ethanol, and we could achieve the efficiency of 89.84, 98.01, 98.28. So the cyclic quadratic graphs you can see, and potential versus current we mapped, and again we can vary the effect of concentration, and we can have of NaOH, formic acid, methanol, ethanol, and we found that electrooxidation is concentration dependent, and the NID current density is proportional to the concentration of the fuel cell. So as sensors, we prepared many sensors, electrochemical based sensors. Initially, we started with simple sensors. This is a very famous that glucose to gluconic acid, and we at that time, Nephune was not available, and then there was a problem. So we used BCA, butyl carbazide acid. So this is also a very useful binder. And using this gold electrode, your particles, we could bind on the surface, and you can create a sensor. And that way we created many sensors. Like this was one, the gold, having zinc oxide nanoparticles on gold electrode also, and we could find the detection of the hydrazine. And we achieved 90.17 microampere sensitivity, and detection limit was very high. This was 0.142, very low. I mean, very high means very small amount we could detect. So here we use cyclic voltammetry and amperometry to understand and to see how this reaction is going on. Similarly, zinc oxide we use for the creating sensors for 4-nitrophenol, and there again similar procedure we applied, and I can show the table there. We had this zinc oxide, iron oxide, the cadmium sulfide, zinc sulfide, all such particles was created, and we found the detection limit and the sensitivity and the compounds for which they were used. All compounds are not good for the environment. We need the sensors to detect as low as their concentration possible, and that can be done using this electrochemical methods. So you can see in some cases the sensitivity was very, very good, and detection limit also was very good. So just to summarize that we could have this metal nanoparticle formation using only a metal complex, which acts as a metal ion source as well as a particle stabilizer. We can find that no contamination there, no stabilizers required, no capping agent is required. Then in microwave, we have time efficient missile methods for nanoparticle preparation using plant extracts. We don't need not to use harmful chemicals, and there are green methods. You have to use minimum energy, that is which is required, and then particles can be can be used as catalyst and for other things which I have told you as sensors, as in fuel cells and so on. So not many have work is being going on in the laboratory and new ideas. Whenever new students come, they have different ideas. So all these things are continuing. Recently, we have a interaction with a US-based startup. They want to, we also worked on, I have not shown there, we also created inclusion complexes with cyclodextrin. So they are interested in our study. They want to use, they want to create inclusion complexes for their food grade materials. So they have given a small project to us and we are working on that. So these, they are using, this is for chocolates, chocolates which are very popular in US and Europe, energy enhancer chocolates we call them. So they use such materials for in those chocolates, and that can be done in the form of inclusion complexes. So they approached us and we are working on that. So inclusion complexes of those materials we can create and they can be very useful in the chocolates. So such, new ideas are very important. And I think when you are working, you should encourage your students to have a broad view. Their vision should be broad. Whatever they want to attempt, they should be allowed to do that work. And it should not be that spoon feeding. If you give liberty to the students, I think they bring to you some results which you are also sometimes surprised that such a interesting results are there. So I give my students a lot of liberty and they do wonders. So it is all the work done by all the students. Teacher is just a manager. He will guide them. He will tell them, okay, do this way, that try it this way, that way. But it is the hard work of the students which brings the fruitful results. So this is not a very fresh photograph. This is an old photograph, but you can see group is quite large, but you'll see mostly female power. Women power is there, and they are the backbone of my group, and they do a lot of work. Although now I have few male members also, male students also, but that time only females were there. So all powerful women, powerful students, female students, they have done wonders, and they are doing wonders now also. After many of them already left, one is in Canada, and this work which I have told you, and she is also involved in that. One is in a company, Varnika, she is in a company in Bangalore. This company is working in nanotechnology, and they are selling nanoparticles and creating processes for nanoparticles. She is doing very good. She gave test there, and the owner of that company was surprised. She got out of 199 marks, and the person was so surprised, and immediately he offered her a job in the company, and she has joined recently, and she is doing excellent. So, others also are working, and the one is a postdoc, two of us postdoc, and they are now doing good jobs, and they are still in touch with me, and we are doing collaborative work also. So thank you very much for patient listening, and now the queries, questions, I will be happy to answer. Thank you. Thank you, everyone.
Sir, I would like to sincerely salute your being so benevolent and compassionate and your commitment to your work ethics. Despite the situation that your wife is COVID positive, even I salute to your wife also, sir, that you didn't postpone and you very well maintained the timetable, whatever we have planned. So before I conclude the informations or knowledge given by Professor Mehta, so students are invited to type their queries in the chat box. If I would like to say that they must interact with Professor Mehta, because this is a rare opportunity and you should get benefit of by him. So in the meantime, I conclude Professor Mehta's informations with you. So you told about various nanostructures using carbon quantum dots, silver sulfide, and mechanism of inhibition, even for viruses, their inhibition of viruses, especially the coronavirus also. He has enlightened us, and the use of mask, the mask composition, and even shared his work with his group regarding these mask composition, and also made us aware about the nanovox mask prepared by his team, available on Amazon, and other functionalized various methods to synthesize functionalized metallosurfactants and alternate to capping agent that is used in case of nanoparticles, covering microwave irradiation, simple synthetic approaches for zinc oxide, iron oxide, and silver nanoparticles. Even, sir, I was thinking that you had not left any area unescaped or untouched in your research. And I was thinking about that you have worked in environmental studies, as well as in sensing, in biological studies, as well as in you can say, in the last I was thinking that only energy area is left, but during your last some slides, I came to know that you have worked in fuel cell also, regarding this electrochemistry field. So it is proud. It gives me a proud feel that we have such an great personality and eminent personality with us. So after that, you told us about the various gel techniques and synthesis of hydrogels and other gel techniques, which are you made us aware about the this metallosurfactants and colloids, and this you can say ki, homogenization, characterization techniques, and use of drug delivery approaches, and the various synthesis, and even advanced techniques also you have made aware. So I would like to request the students also, as well as my faculty colleagues also, if they have some discussion, they want some discussion with Professor Mehta, they are most welcome.
Excuse
And
sir.
Yes, please.
मैम आपने कंक्लूड कर दिया मैम?
नहीं, अभी मैं
Okay,
कर रही हूँ.
Okay,
हाँ,
ma'am.
हाँ.
Please
So
continue.
so I also I would like to thank my organizing team members also, Dr. Sumit Kumar and Dr. Krishna Kumar, for organizing in such a nice way these lectures. Sir, this was our third lecture of this first lecture series, online lecture series. However, I can assure you that certainly we will be having you in among us in conventional mode also, after the situation will be in control. So now I would like to invite Professor A.K. Sharma to share some words and some thanks with you and with all the listeners. Professor Ashok Sharma.
Good evening, Professor Mehta.
Good evening, sir.
And all the participants in this webinar, in this lecture. Sir, thank you very much for sparing time and enlightening all of us with your lecture. It is always a treat to listen to you. However, due to this pandemic issue, we could not invite you and interact with you personally. We were not knowing, sorry for that respected madam has been infected with this corona. Despite that, you delivered your scheduled lecture. This shows your discipline and commitment to your duty. As madam herself is a doctor, so definitely she will recover very soon, I'm sure. We all wish a speedy recovery to her, and please convey my personal wishes and regard to her also. Due to this pandemic, the visit planned by me could not materialize to your university and this sophisticated instrumentation facility.
Yeah.
I think, Madam, if you allow, then our students can write their queries to us in the chat box or in a mail to us, and we can send that queries to Professor Mehta, because he is already busy and already too much time he has spent with us. So that will be a nice idea.
Sure. Why not? Mhm.
Thank you, Dr. Saab. Thank you for nice words.
And even सर प्लीज एक सेकंड सर
please.
एक इम्पोर्टेंट बात रह गई थी कि जो अपने स्टूडेंट्स को एक मैसेज देना चाह रही थी कि भी जो आपका जो चंडीगढ़ रीजन के अराउंड जो क्लस्टर आपने बनाया है,
हाँ जी।
तो आई थिंक मेरे स्टूडेंट जो डिपार्टमेंट के हमारे डिपार्टमेंट के हैं वो उस क्लस्टर में मेंबरशिप ले सकते हैं।
हाँ, श्योर श्योर, व्हाई नॉट।
ओके, तो ये मैं अपना आई एडवाइज माय स्टूडेंट्स दैट दे शुड गेट मेंबरशिप टू दैट क्लस्टर around Chandigarh region and all other clusters also. And I wish for ma'am to her soon recovery. And now I would invite Professor Dr. Sumit Kumar to conclude the and thank. And I think some questions are also, one or two questions are also there in the chat box.
Yeah, please, please.
Thank you, ma'am. Sir, first of all, I would like to thank you for a nice lecture, sir. There are two questions from our students. One is that what is the difference between size calculated from XRD and SAM, and which is more accurate?
Okay. Actually, SAM doesn't give exactly the size, but TEM gives the exact size, right? So the size by TEM is more accurate as compared to, because in the XRD this relation which we use, there are many assumptions. So therefore, the actual size is given by TEM.
And other question is that, sir, what is the respiratory safety regarding the nanoparticle incorporated masks?
That is a very important question, because the toxicity is one very important parameter. Then if we inhale the nanoparticles, they will be naturally very damaging. Once they go into the body, they can enter into any cell and then can damage. So the quantity of nanoparticles which are being used in this formulation, for example, which we have created, the particle is in the quantity which is nearly same to the particles which are available in the atmosphere, right? Okay. Which are not harmful to ourselves. So small quantity is not harmful. So the limit is not crossed, so that the particles which are embedded on the mask, they do their function. But even if they go inside our body, although we should not allow them to go into the body, but even if they go in, sometimes it may, but then they are not harmful.
Okay, thank you, sir. Just as a curiosity, I also have one small query, sir.
Yeah, please.
So you have done so many morphological studies, different sizes and different shapes, basically.
Yeah.
So is the nanoparticle shape is related to some particular activities, or they are not dependent on shape with respect to their biological activity or other type of their
biological activity? Playing with the parameters, temperature, concentration, and all these parameters, there you can create the shape.
Yeah, we can create the shape. My question is that, sir, does the shapes impact their application?
Okay, application wise. Okay. Although we consider spherical particles as the best particles, okay, for biological purposes, because you have to take the drug inside, And
that I,
particles are spherical, and but only thing is that biological component which you are using, you have the attraction of that particle with the spherical particle or with the other shape particle is better. That you have to see. If it is better with the, for example, cylindrical particles or other shape particles, then we have to use that shape particles.
okay. Thank you very much, sir.
Okay.
So it's honor to listen you, sir, and it's my privilege to thank all the audiences, Professor Ashok Sharma, Professor Sumunata ma'am, my colleague Dr. Krishan Kumar, and my other colleagues from the department, Dr. Hariom, Dr. Sonia, Dr. Dinesh, and Dr. Rajender. And I am also thankful to all the students who have listened here. And some of our students were listening you live on the YouTube also, sir. And special thanks to Mr. Kuldeep from our computer department who has organized this meeting and did it live on the YouTube. And we all are thankful to you and wish you a speedy recovery for your wife and a safe time for you ahead, sir. Thank you very much, sir.
Thanks a lot. Thank you very much. Sharmaji, thank you very much.
Thank you, Ji. Thank you.
Thank you,
Thank you, sir. Thank you very much.
My wife remembers Sharmaji a lot sometimes, so so I will convey madam and all of your wishes to her. And although she herself was involved in, taking the employee of her university to hospital for vaccination. So I don't know where from she got this, but anyhow, she is brave enough to counter this, I think so.
Definitely, sir. She will recover very soon.
Yeah.
I know she is very strong.
Yeah. She told me that you
Sure,
must
sir.
take lecture. There's no problem. I'm okay.
Sir, special thanks to ma'am. Thank you.
sir.
Sir. Thank you.
Thanks a lot.
Okay, namaskar.
Thank you very much.
Thank you, everyone. Thank you.
Thank you, one and all. Thank you all.
Thank you.